Chapter 13, From Matter to Information to Matter, Defining Genetic Information. Genetic information refers to the hereditary instructions encoded in the nucleic acids of an organism, dictating its development, functioning, and traits. It is a complex concept that embodies both structural and functional dimensions, intricately linked to the molecular biology of life. Defining genetic information necessitates understanding its physical embodiment, replicability, and the cellular context in which it operates. The primary medium of genetic information is nucleic acids, DNA and RNA. These molecules uniquely qualify as bioinformation due to their dual characteristics of being physical entities and inheritable. Replicability is a cornerstone of genetic information, as it ensures the continuity of life across generations. Central to this replicability is the origin of replication, a specific sequence on the nucleic acid that the replication machinery recognizes and uses to duplicate the genetic material. A nucleic acid molecule outside a cell, devoid of a replicative system, is merely matter. Even a complete genome or chromosome loses its essence as genetic information when isolated from the cellular environment. For a DNA molecule introduced into a cell to qualify as genetic material, it must integrate into a chromosome or be associated with a functional origin of replication. This inherent requirement for replication ties genetic information to a replicator, emphasizing its dependency on the cellular milieu. The functional aspect of genetic information is encoded in stretches of nucleic acids that fulfill specific roles. These include coding regions that produce RNA molecules such as mRNA, tRNA, and rRNA, regulatory sequences like promoters and enhancers, and structural elements involved in gene regulation or replication. Importantly, even genomic regions with currently unknown functions may hold genetic information, highlighting this concept's dynamic and evolving nature. Defining the minimal unit of genetic information adds another layer of complexity. A single nucleotide within a protein-coding gene, though seemingly insignificant, can drastically alter the encoded protein sequence and function if removed or modified. Thus, every nucleotide associated with a functional genetic system can be considered the minimal functional unit of genetic information. This extends to non-coding regions and accessory genetic elements, as they contribute to the overall expression and regulation of the genome. The cellular environment is indispensable for the expression of genetic information. Despite being autonomous entities, viruses, plasmids, and viroids require a host cell to replicate and express their genetic instructions. This reliance underscores the emergent nature of genetic information, which becomes functional only within a replicative and cellular context. In essence, genetic information can be defined as the functional, inheritable bioinformation encoded in nucleic acids, DNA or RNA, that resides within a replicable structure and is capable of expression within a cellular environment. Each nucleotide within such a system constitutes the fundamental unit of genetic information, emphasizing its intricate relationship with life and the cellular processes that sustain it. Organization of genetic information. Genetic information is a fundamental characteristic shared by all propagating entities. This includes a cellular propagating entity such as viruses, plasmids, and viroids, as well as cellular organisms like prokaryotes and eukaryotes. Even subcellular entities like mitochondria and chloroplasts contain genetic material. However, the organization of this genetic information varies significantly across these entities. In cellular organisms, genetic material is primarily composed of double-stranded DNA. In contrast, viroids are made of RNA, while plasmids typically consist of double-stranded DNA, although some are single-stranded. Viruses exhibit a wide range of genetic material with either DNA or RNA that can be single or double-stranded. Most cellular entities, including bacterial genomes, contain circular double-stranded DNA, although some bacteria possess two circular chromosomes. Eukaryotic organisms, on the other hand, have genomes composed of multiple linear double-stranded DNA molecules. Subcellular entities like mitochondria and chloroplasts usually contain single circular DNA molecules, though exceptions exist with multiple linear DNA molecules. The genome size tends to correlate with the organism's complexity, with viroids and plasmids having the smallest genomes, followed by viruses, bacteria, and eukaryotes. Genomes consist of genes, regulatory elements, structural components, and sometimes non-coding DNA, often referred to as junk DNA. Eukaryotic organisms are typically diploid, meaning they possess 2 copies of each chromosome, while haploid cells contain only 1 copy. The human genome, for instance, consists of 46 chromosomes, 23 pairs. The total length of DNA in a human cell, if stretched end to end, would measure around 2 meters. However, it is tightly packed into a nucleus measuring 10 to 15 micrometers diameter. Each genome includes origins of replication, specific nucleotide sequences that are recognized by proteins to initiate DNA replication. Without these origins, DNA would not be inherited. Essential genes for replication, gene expression, transcription and translation, and regulation are common across all cellular organisms. Metabolic genes help generate energy and synthesize biomolecules, while others manage material exchanges between the organism and its environment. In multicellular organisms, cell differentiation involves the selective activation or repression of genes, creating distinct phenotypes in different cell types. Thus, the genome functions as a toolkit with an instruction manual guiding when, how, where, and to what extent each gene should be expressed. Genomic emergence, multistrata emergence in genetic information. The organization of genetic information and its influence on life processes can be understood through a multi-level system of emergence, where each level builds upon the previous one. Starting from the most fundamental components, this hierarchy progresses through increasingly complex levels, culminating in the competent, functioning organism. Level 1, genetic information and genes. Genes, the functional units of this information, are diverse in their roles. Structural genes encode proteins that form cellular components. Regulatory genes control the expression of other genes, and metabolic genes direct enzymes for energy production and synthesis. These genes collectively form the genetic blueprint for all living organisms. For example, the gene for hemoglobin enables oxygen transport in red blood cells, while the insulin gene regulates blood sugar levels. Level 2, functional gene products. At this level, genetic information is translated into functional products such as proteins and RNA molecules. These products act as the machinery of life, facilitating essential processes. Enzymes catalyze biochemical reactions, structural proteins provide cellular support, and regulatory proteins control cellular activities. The glycolytic enzymes that break down glucose for energy exemplify this level, as do histones that compact DNA into chromatin. Level 3, interactions among gene products, leading to phenotypes. The third level involves interactions among gene products to produce observable phenotypic traits. A phenotype arises not from a single gene, but through the concerted actions of many gene products. For instance, muscle contraction results from the interaction of actin, myosin, and associated proteins, while skin pigmentation depends on the activity of enzymes like tyrosinase in the melanin production pathway. Such interactions are integral to the specialization and functionality of different cell types. Level 4, cellular phenotypes, forming organ functions. At this level, the phenotypes of individual cells combine to create tissues and organs with specific functions. Specialized cells collaborate to perform the complex tasks required for organ functionality. For instance, hepatocytes work together in the liver to detoxify chemicals, synthesize proteins, and produce bile. Similarly, heart muscle cells coordinate to enable rhythmic contractions that pump blood throughout the body. The integration of cellular phenotypes results in organ-level functions essential for the organism's survival. Level 5, organ functions and organismal fitness. The fifth level involves integrating organ systems, each contributing to the organism's overall fitness. Organ functions do not operate in isolation. They work collectively to support survival, reproduction, and adaptation to environmental challenges. For example, the respiratory and circulatory systems work in tandem to deliver oxygen to tissues, while the immune system protects against pathogens. The efficiency of these organ systems determines the organism's ability to thrive and reproduce, directly influencing its fitness. Mendelian inheritance. Gregor Johann Mendel was a monk and is considered to be on par with Darwin and Mendeleev when it comes to scientific thinking. In fact, Mendel was a contemporary of Darwin, although somehow Darwin was unaware of Mendel's work. Mendel conducted experiments, recorded his observations, and ultimately proposed the laws of inheritance. Mendelian inheritance describes the principles of trait transmission. Mendel's experiments with pea plants laid the foundation for our understanding of this concept. Mendelian genetics includes the law of segregation, which states that each organism carries two alleles for a trait, one from each parent, and these alleles segregate during reproduction. The law of independent assortment states that different traits are inherited independently as long as they are on different chromosomes. Additionally, dominant alleles manifest their effect in the phenotype even with only one copy, while recessive alleles require two copies. Mendelian inheritance provides a predictable framework for understanding trait transmission and serves as the basis for classical genetics, which has been further developed with advancements in molecular genetics and the study of complex inheritance patterns. Mendel's brilliance went unrecognized for a long time. On February 8, 1865, Mendel presented his work to the Brunn Society for Natural Science. His paper, Experiments on Plant Hybridization, was published the following year. While his work was appreciated for its thoroughness, its importance was not understood then. Mendel's work was too ahead of its time and contradicted popular beliefs about heredity. "My time will come," Mendel once said, but it took over 30 years for his work to be appreciated. Mendel should have written a book too. Mendel's experiments and formulation of Mendelian inheritance principles went unnoticed during his lifetime, but gained recognition posthumously. His work was rediscovered and became the foundation of modern genetics, leading to significant advancements in our understanding of genetic inheritance. Mendel's principles have profoundly impacted various fields, including biology, medicine, and agriculture. Three botanists, Hugo de Vries, Carl Correns, and Erich von Tschermak, independently rediscovered Mendel's work in the same year and credited him as the first to discover the laws of inheritance. My scientific studies have afforded me great gratification, and I am convinced that it will not be long before the whole world acknowledges the results of my work. Gregor Mendel. Maintenance, expression, and regulation of bioinformation, genes alone cannot result in a phenotype. They must be part of a system with the necessary gene expression components. This means that genes needed for transcription and translation must be present to express themselves and other genes in the genome. Replication is essential for temporal existence, ensuring continuity across generations. Gene expression is the actionable process that actualizes this temporal existence by making things happen. While serving as the repository of information, the genome cannot achieve anything by itself. Genetic information represents only the potential for temporal existence, whereas gene expression is the mechanism that transforms this potential into reality. Although these processes involve many enzymes, we will focus on the flow of information. Replication and cell division, genetic continuity. The essence of life on Earth is propagation or genetic continuity. Replication is the key process that enables genetic continuity. As discussed in the previous chapter, life is defined by the presence of inheritable information, and this inheritance is only possible through replication. DNA replication is catalyzed by the enzyme DNA polymerase, which is a DNA-dependent DNA polymerase. DNA polymerase depends on a template, meaning it adds deoxynucleotides one base at a time in a specific order based on the complementarity of the template strand. This dependency on a template is essential for the inheritance of sequence information. Without it, random sequences would be generated, which is not a form of information inheritance. The two strands of parental DNA separate at regions known as the origin of replication. However, DNA polymerase cannot initiate replication on its own. It can only add to existing nucleic acid strands, polymerizing the DNTPs to the 3'OH end of DNA or RNA. Therefore, replication begins with the synthesis of a short stretch of RNA strand, primer, by a dedicated RNA polymerase called primase. DNA polymerase then takes over and adds DNTPs until the end of the strand. Two strands are synthesized simultaneously, one continuously and one discontinuously, at the replication fork. Eventually, two new double-stranded DNA molecules are formed from one original double-stranded DNA molecule. This is known as semi-conservative replication because each daughter DNA molecule shares one strand with the parent DNA molecule. We have detailed knowledge of how DNA replication occurs in bacteria, eukaryotes, viruses, and plasmids, although these details are not discussed here. The important point to note is that replication is a highly efficient process in terms of speed and accuracy. Information is copied with remarkable accuracy based on the Watson-Crick base pair, with an error rate of 1 mistake per 100,000 nucleotides polymerized. Furthermore, dedicated repair mechanisms scan the newly synthesized DNA for mismatches and other errors. DNA polymerases can add about 1,000 bases per second, making them incredibly efficient. However, errors are inevitable, and changes in nucleotide sequence, known as mutations, will occur. Generally, replication precedes cell division. Each daughter cell receives one of the two double-stranded DNA molecules formed. There are two types of cell divisions, I, mitosis, and II, meiosis. Mitosis is the type of cell division in which the daughter cells are identical to the parent cell in terms of the quality and quantity of genetic material. Somatic cells in humans have 23 pairs of chromosomes, including the X and Y chromosomes. This is because we are biparental, meaning we inherit one set of chromosomes from the mother, maternal set, and another from the father, paternal set. The daughter cells will each inherit identical copies generated through replication of the genomes of the parent cell. All of the somatoplasm, which is the entire body except the gamete-generating cells, is achieved through repetitive mitosis. Therefore, the genome of all somatic cells is identical, except in some blood cells and cells with mutations that may have occurred. In meiosis, there are qualitative and quantitative differences in the genomes of the daughter cells relative to the parent cells. Even among the daughter cells, there are qualitative differences. The qualitative differences occur because of recombination between the non-sister chromatids of homologous chromosomes. As a result, there are new combinations of alleles within homologous chromosomes at the molecular level. Secondly, different combinations of chromosomes are segregated independently, forming combinations of maternal and paternal chromosomes. As a result, the genomes of the daughter cells are qualitatively different from those of the parent cells. The genomes of the gametes will contribute to the somatoplasm of the next generation upon fertilization. In a way, all of our somatoplasm is striving to make the somatoplasm of the next generation only. The next generation will live with the somatoplasm that we made, and they will make new gametes that will make up the somatoplasm of the next generation. When we talk in terms of genomes, we are referring to the combinations of genes and their alleles. We inherit these genes from our ancestral humans, which have changed slightly. Gene expression. If you do not express who you are, you do not exist. The central dogma of molecular biology encapsulates the flow of genetic information within a biological system, describing how information in DNA is transcribed into RNA and subsequently translated into proteins. This framework provides the foundation for understanding how the instructions encoded in the genome are expressed to sustain life. At its core, the central dogma establishes the directional flow of information. DNA serves as the template for RNA synthesis, transcription, and RNA guides protein synthesis, translation. Proteins, in turn, act as the functional and structural molecules of the cell, driving biochemical reactions and forming the architecture of life. At the first level, DNA replication ensures the faithful inheritance of genetic material during cell division, maintaining continuity across generations. Transcription, the second level, involves the synthesis of messenger RNA, mRNA, from the DNA template, a process mediated by RNA polymerase. The third level, translation, converts the nucleotide sequence of mRNA into a polypeptide chain using ribosomes and transfer RNA, tRNA, to decode the genetic instructions. While the central dogma represents a unidirectional flow, exceptions exist, such as reverse transcription in retroviruses. Nevertheless, the central dogma underscores the elegant simplicity of life's molecular machinery, linking the static genetic blueprint to dynamic biological functions and evolutionary adaptability. Gene regulation. Regulation is everything. Differential gene expression refers to the phenomenon where different genes are expressed at varying levels or in different cell types, tissues, or developmental stages. It is a fundamental process that underlies the diversity and complexity of living organisms. This regulation of gene expression allows cells to respond to their environment, carry out specialized functions, and maintain overall organismal homeostasis. The regulation of gene expression is controlled by a variety of molecular mechanisms. At the transcriptional level, differential gene expression can occur through the binding of specific transcription factors to DNA regulatory regions, such as enhancers or promoters, which either enhance or repress gene transcription. Epigenetic modifications, such as DNA methylation and histone modifications, can also play a role in regulating gene expression by influencing the accessibility of DNA to the transcriptional machinery. Post-transcriptional regulation mechanisms further modulate gene expression by controlling mRNA stability, alternative splicing, and translation efficiency. Additionally, microRNAs and other non-coding RNAs can interact with mRNA molecules to fine-tune their expression levels. The consequences of differential gene expression are vast and have profound implications for cellular function and organismal development. Differentiated cell types, such as neurons, muscle cells, or skin cells, arise from precursor cells through specific gene expression patterns. During embryonic development, precise temporal and spatial gene expression regulation directs tissues and organs formation. Dysregulation of differential gene expression can lead to various diseases. For example, cancer often involves aberrant gene expression, where oncogenes are overexpressed, and tumor suppressor genes are silenced. Gene regulation plays a pivotal role in orchestrating biological processes, analogous to the precise control of tools in a toolkit. It determines how and when genes are expressed, uncovering the complexities of an organism's functioning. This regulation ensures that genes are used judiciously, contributing to the dynamic equilibrium within the biological system. Epigenetic regulation. Epigenetic regulation is a cornerstone of gene expression control, allowing organisms to respond dynamically to environmental and developmental cues without altering their genetic code. While gene expression in bacteria relies on straightforward mechanisms like activators, repressors, and RNA molecules, multicellular organisms require complex regulatory systems to manage the diverse functions of various cell types. Epigenetic modifications are essential in this context, providing a sophisticated layer of control over the genetic blueprint. Epigenetics refers to heritable changes in gene expression that occur without modifications to the DNA sequence itself. This regulatory system modifies the accessibility and functionality of genetic material through covalent or non-covalent changes, determining when and how genes are expressed. Epigenetics employs a variety of chemical alterations that collectively fine-tune gene activity. DNA methylation, a key mechanism, involves the addition of methyl groups to cytosine residues, typically resulting in gene repression. Histone modifications, including acetylation and methylation, play dual roles depending on the specific chemical group added and the residue modified, either activating or repressing gene expression. Other mechanisms, such as histone phosphorylation, ubiquitination, and chromatin remodeling, further contribute to the intricate regulation of genes. Non-coding RNA molecules, including microRNAs and long non-coding RNAs, provide additional layers of post-transcriptional control, while genomic imprinting ensures allele-specific gene expression based on parental origin. These modifications work in concert to regulate gene expression, cellular differentiation, and environmental responsiveness, forming the basis of tissue specialization and organismal adaptability. Epigenetic modifications and their functional impact. Epigenetic changes represent chemical alterations in DNA or its associated proteins that can either activate or repress genes. These modifications can be stable enough to pass to subsequent generations or reversible, allowing adaptation to shifting environmental conditions. Crucially, they are instrumental in processes such as development, cellular differentiation, and environmental responses, determining which genes are active or silenced in specific cell types. Research into epigenetics has deepened our understanding of development, disease mechanisms, and the gene-environment interaction. DNA methylation and histone modifications, for example, have highlighted how nutrient availability, stress, and environmental pollutants shape cellular phenotypes without necessitating genetic mutations. Such insights hold significant promise for advancements in medicine, agriculture, and environmental science, demonstrating how external stimuli can influence health and disease susceptibility through epigenetic pathways. Influences on epigenetic patterns. Various factors influence epigenetic changes, including diet, circadian rhythms, stress, and parental contributions. Nutrients like folate and vitamin B12 modulate DNA methylation, impacting cellular functions. Sleep disruptions affect histone acetylation, influencing metabolic and stress-related genes. Parental epigenetic marks established in germ cells can alter offspring traits, as seen with maternal diets affecting fetal gene expression. Environmental stressors, including toxins and psychological pressures, further drive epigenetic changes, with chronic conditions often arising from these alterations. Social hierarchies also shape epigenetic patterns. For instance, dominant animals exhibit reduced methylation in genes linked to reproduction and aggression, while subordinates display increased methylation in stress-related genes, leading to distinct physiological and behavioral profiles. Though reversible, these reciprocal modifications highlight organisms' adaptability to their social and environmental contexts, underscoring the ecological significance of epigenetic regulation. Generational and transgenerational epigenetics. Epigenetics has profound generational implications, as demonstrated in the Dutch hunger winter, where malnourished mothers produced offspring with altered glucose metabolism. Intergenerational effects, like stress-induced methylation changes in Holocaust survivors' children, exemplify how epigenetic marks can influence immediate descendants. Transgenerational epigenetics extends this phenomenon across multiple generations, with animal studies showing stress or toxin exposure leading to lasting epigenetic modifications. The concept of adaptation in biology has traditionally been conflated with genetic evolution. However, adaptation is more accurately understood in an epigenetic context as a non-genetic responsive process occurring within an organism's lifetime. Epigenetic mechanisms provide organisms with reversible and conditional traits that enable them to adapt dynamically to environmental pressures, offering a nuanced perspective on evolution and adaptation beyond genetic mutations. Genomic imprinting. Genomic imprinting is a fascinating, complex, and intriguing phenomenon in genetics. Certain genes are expressed differently depending on whether they are inherited from the mother or the father. All the organisms that reproduce through obligate sexual reproduction are biparental, i.e., contain diploids with one maternal set and one paternal set. Some genes are imprinted, while most of the genes are not. Some imprinted genes are expressed only from the maternal set, while others are expressed from the paternal set. Examples of imprinted genes include IGF-2, expressed from the paternal allele to promote fetal growth, and H19, expressed maternally to regulate growth. UB3A, maternal expression, and SNRPN, paternal expression, are crucial for brain function, with disruptions causing Angelman and Prader-Willi syndromes, respectively. KCNQ1 impacts heart development linked to imprinting disorders. All the cells in our body, except for the germplasm, are diploid, 2N, meaning that we have two copies of each chromosome, one from each parent. The imprinted genes in all of our cells bear the signatures of their origin, either from the maternal or paternal set. Imprinted genes have one copy that is silenced based on its parental origin through epigenetic modifications, which are chemical changes to DNA that do not alter the underlying sequence, but affect gene activity. The main mechanism is DNA methylation, where small chemical groups, methyl groups, are added to specific regions of DNA called imprinting control regions. Imprints are epigenetic marks established during gametogenesis, erased and reset in each generation. During germ cell development, parental imprints are erased to create a blank slate. As germ cells mature, sex-specific imprints are established, maternal imprints in oocytes and paternal imprints in sperm, via DNA methylation in imprinting control regions. After fertilization, imprints are maintained throughout embryonic development and in somatic tissues, ensuring proper gene expression. Despite global epigenetic reprogramming in early embryos, imprints are protected, preserving parental-specific expression. This dynamic erasure, establishment, and maintenance cycle allows genomic imprinting to perpetuate across generations while adapting to specific parental roles. From an evolutionary perspective, genomic imprinting is thought to have evolved due to conflicts between maternal and paternal interests, particularly in species where offspring compete for maternal resources. This is known as the parental conflict hypothesis. For example, paternal genes may favor faster growth to ensure offspring survival, while maternal genes may regulate resource allocation to balance care for multiple offspring. Imprinting, therefore, reflects a fine-tuned balance shaped by evolutionary pressures. I remain unconvinced as to evolutionary explanations of genomic imprinting so far. I speculate that the primary function is to prevent the fusion of gametes of the same sex, that is, the fusion of two sperms or two eggs, giving rise to uniparental zygoids. These abnormal zygoids die early in development due to abnormal gene expression of the imprinted genes. Therefore, by preventing such zygoids, I think genomic imprinting ensures reproduction only through fertilization, fusion of male and female gametes. I suspect a link between genomic imprinting and sexual reproduction. All chromosomes have sex marks, either maternal or paternal, in the form of epigenetic marks, which are imprinted genes.